Hello everyone, and welcome to today's webinar titled Panel Presentation, Mitigating Variability in Somatic Variant Interpretation. This webinar is part of the 12th Annual Precision Medicine, Genomics, Genetics, and Molecular Diagnostics virtual event series in 2024. I'm Caitlin McCann-Jankovitz, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by QIAGEN Digital Insights. For more information, please visit www.digitalinsights.qiagen.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. Now, let's get started. Our speakers today will be Jenni Fairley, Deputy Scheme Director, Molecular Pathology at GenQA, Kathryn Bungartz, Professional Clinical Solutions Manager for QIAGEN Digital Insights, Sheryl Elkin, Chief Scientific Officer for QIAGEN Digital Insights Oncology, Richard White, Associate Professor, Physician, and Scientist at Memorial Sloan Kettering Cancer Center in the University of Oxford, and finally, human geneticist Jim Hayes. For a complete biography on our speakers, please visit the Presenter tab from the menu on the left of your screen. And with that, I'd like to turn it over to Kathryn Bungartz to get us started. Take it away, Kathryn.
Thank you for the nice introduction, and thank you everybody for joining. We hope to have a lively discussion today. We're gathered to discuss how software can be used to assist with somatic interpretation, and what are the causes of the inconsistency interpretation, and how we might mitigate, perhaps mitigate this inconsistency with a high quality knowledge base. Well, I'd like to start off the discussion by briefly outlining the format of the recently published study comparing variant calls using a clinical decision support software, comparing this with methods of different laboratories. And Jenni, if you could take us through this and maybe also point out what you see as the primary take-home message or messages.
Kathryn. So to perform this study, sorry about this. We went out to some laboratories asking them to help us out with it, and we went to expert laboratories where we got them to send us through variants in the form of VCF files that they had found in their clinical practice. And then we went further with that, and sorry about this. And we sent them out to the eight independent laboratories, who then sent them back to us as anonymized, and we randomized them and sent them out amongst the other laboratories, and at the same time uploaded them to QCI1 software to do variant analysis. And across, we had a total of 77 different VCFs submitted across both heme and solid tumours, and that represented more than 200,000 different variants. And what we wanted to do was compare the tiering according to AMP tiering classification across the different variants between both QCI and the different laboratories. So each set of variants were looked at by QCI and either one or two different laboratories. And each laboratory nominated an expert to sit on a panel to discuss any differences between the variant classifications. And we needed that expert panel because currently there isn't any real gold standard for somatic variant classification. Laboratories have their own methodology for doing it, and there isn't any one way that everyone is using. There are lots of different ways to get to hopefully the same classification. However, one thing that I think the ultimate thing we did find with the study is there are still differences in the classification of variants. And that's true between different laboratories, i.e. different individuals doing the classifications. And there were differences also between the classification of variants between individuals and QCI. However, the number of differences was not huge in terms of the actual clinical classification outcomes in the end. And there were probably as many differences, there were actually more differences between the individual laboratories than there were between the panel observed variants and that determined by QCI1. I think we also observed that there were some differences between different tumour types, so that we could see that there was differences between solid tumours and heme tumours, and some of that was to do with the way laboratories were approaching the classification. Sheryl, I don't know if you want to come in here and comment on that as well.
Sure. I have so much to talk about. We found it really fascinating. The whole project was really interesting. We were blinded to the laboratories that were engaging in the study, but we did a lot of, at the end, once the data was collected, we did a lot of analysis together of what were the differences and what caused the differences. And one of the things, if you bring up the difference between, the variability between solid tumors versus hematological cancers, and at first glance, the variant interpretation itself, in terms of what is the biological impact of this variant, was not more in heme versus solid tumors or the other way around. There may be more novel variants in heme which cause more changes, but what we actually found in this study, the total number of differences, I think, for was slightly smaller for heme than it was for solid tumors, but the numbers overall were very small. I think it was 9 total were different in solid tumors and 5 that were different for the heme tumors out of the entire data set that we determined were actual differences between the expert panel and the QCI determination. One of the things I was thinking about, the solid tumor number in this case was actually slightly higher. Of course, when you're dealing with numbers under 10, it's hard to make a big conclusion about whether one is easier than the other. With the AMP tiering classifications, when you're looking at the impact of the level of evidence for a gene or a variant within a gene in a disease, a lot of what you're looking at for solid tumors is impact on a drug availability. So, a tier 1A is going to be something that has an approved drug that can be used to target that alteration. And a tier 3 is going to be VUS, and tier 2 might be something in clinical trials. So those are really clear-cut delineations that can often be assigned to drug labels and dependent on the disease. Whereas with heme tumors, you're dealing with prognosis and diagnosis and levels of evidence for prognosis and diagnosis, which is a lot more subjective to delineate the difference between a tier 1, a level A and a level B, or a level B and a level C, which is the difference between tier 1 and tier 2. And I think Jenni would agree that one of the comments that we got from the expert panel was that these differences between tier 1 and tier 2 and level A and level B, or level B and level C, were not super clinically relevant as long as the variant was itself reported. So I could stop there. I could go on for the whole hour. But I won't.
So maybe I can just quickly kind of summarize that the conclusions of the study were basically that there is, in fact, there was found to be quite a high level of concordance between the lab's decision and the software. And maybe another conclusion is that the discordance, that the small amount of discordance that was there actually even exists between experts. So not all experts agree with how to call a variant. I wonder, Rich, maybe you could touch more. We heard some of the reasons maybe for this between solid and heme. Maybe you could touch on, as a practicing oncologist, what you think could be more reasons for discordance or inconsistency in variant calling.
Yeah, sure. I thought one of the interesting results is that, there is disagreement even among the experts. And I think the reasons for that probably have both technical as well as biological, reasons. And I think they're both important to consider, right? One of the challenges from a technical standpoint on variant calling is that as tumors get more heterogeneous, which is definitely the case in solid tumors, the variant calling becomes more complex, right? The software that calls the variant has to make statistical judgments about whether that variant is really enriched. And that, in turn, impacts the way to interpret things, because if you have a variant that the software is sort of like, okay, well, this is present at 4.9% of the reads, right? Is that biologically significant? Well, it might be. And another software might say, okay, well, this is at 5.1%, and therefore it reaches some threshold for significance, right? So I think one level that makes it hard is that for borderline things, there's technical variation, and that can lead to a lot of confusion. Is this a real variant or not, or is it important? Another sort of source of difficulty is that, again, in heterogeneous tumors, for mutations in which it's really clear that there's either a prognostic or let's say, a therapeutic benefit, very often, if the variant is expressed or is it at a pretty low VAF, right? Let's say it's not, it's somewhere in the middle. It's actually hard knowing if that's clinically important. And in the tiering, that could become very important, right? Because you could sort of say, well, this gene might be really important, but it's present at a pretty low VAF, and therefore how biologically important is that? Again, that's a judgment call, right? There's no strict criteria to sort of say, that's the VAF at which it becomes biologically important. And then, so I think the third reason, and I'll kind of pause after this, is actually much more challenging, which is that if you have a gene in which the biological function is well known in a particular context, the problem is applying that same gene to a different biological context or a different cell type can actually be extremely challenging in a lot of cases. Because just because you have a BRAF mutation in one situation, it doesn't mean it's going to do the same thing in another situation. And so I think that that's often where you get into this confusion, not so much between tier 1 and tier 2, but tier 2 and tier 3, where it's unclear what that particular variant might be doing in this particular context out of the canonical ones. So I'll kind of stop there, and I think that there's technical things, how you call a variant, and then the second is what is the threshold for thinking that variant is important in any context, and then the third is what is the likelihood that this variant at a certain VAF is actually important in this particular context. And I think all three of those play into this non-concordance.
Yeah, there's a lot to unpack there. So do we want to, maybe we can start, Jim, with how the study was set up and what you think the role of the parameters, say the secondary analysis or the filtering parameters, might affect analysis.
Yeah, absolutely. And so, Richard, absolutely. Like, you make a great point about the software needs to make decisions on where those appropriate thresholds are and where the cutoff should be drawn. I mean, this is ultimately, it is a binary output. Something is in the VCF or it is not, but it all depends on where you draw the line. And I think when we were setting up this study, that's something where we were trying to keep it unified across the group. So if you go into the methods of the study, you'll see that there were strict parameters that were applied to every sample that was uploaded. Everything was uploaded that had the same allele fraction cutoff, that was in the same gene list. And the software would take a VCF, and so if it was in the VCF, there would be filtering. And those filters were set across the board. And so it could have been the case when somebody would have, their variant caller would have, passed something on with the VAF of 4.9, but if it was a 5.0 cutoff, then it wouldn't actually show up in the software for interpretation. Again, this was an effort to make sure that everything was consistent across the groups, across the 8 different participating sites, but there needed to be a line cut off somewhere. And so I feel like there's an aspect of this study that kind of took away part of that human element, right? This would be where if this was software to be utilized in a local setting, you would know your pipeline, you would know how it would be set up, and you know what thresholds you are comfortable with looking at. And also, you may, decrease the stringency of those thresholds in order to see some of those things that are borderline, and then really leave it up to the human interpretation of, is it relevant biologically? Is it real technically? Going back into the read stack, saying like, is this something that I trust, and is it worthy of actually doing interpretation? I think it's really important that regardless, there is still, as easy as computers and platforms and software can make this and streamline the process, there still really is a human element that is required. Where are these lines drawn? How do we interpret it? And we would love it if it would be black and white across the board, but it's definitely not the case.
Yeah, thanks, Jim.
We had a perfect example of that in the study as well, because we did have some VCFs that had thousands of variants in them, and where this was a source of discordance. But when we looked at those variants, they were all around about the same variant allele frequency, which was less than 10%. So we'd sort of set the cutoff at 10% for those particular VCFs. That discordance would have all disappeared, and we assigned that as a technical issue as opposed to a discordance. So I think that's quite a good example of what Jim's been talking about, like individual cutoffs need to be applied to different samples.
Yeah, I think there's judgment that has to be applied on every level. Both, we made a decision about setting all of the cutoffs to be equal across all the labs so that we would have a standard. But as Jim mentioned, if you're doing it in your own lab, there are going to be differences on how you set the filters, whether you lock them down forever or whether you adjust them usually not on a case-by-case basis. But we wanted to sort of only look at one piece of that when we were, in order to not have this be a 7,000-page paper, but really just looking at the interpretation itself, we chose to not talk about the differences between filtering and really look at the differences in assigned interpretation. And so that was how we came up with, the denominator of about 250 variants that we looked at, where the discordance with QCI was only about 14 of them. And the discordance among the different laboratories ended up being about 30%, which was not shocking, but interesting.
Yeah, and I'll just add one thing that I think is interesting is that depending on where each individual lab does set their filtering, will have big impacts on the biological, the discordance, I should say, because then you start to get into things that might be more questionable, low VAFs and things like that. So making those decisions about where you set the filter has a very real impact on concordance or discordance.
Yeah. So another item that was just kind of briefly covered was the idea that you have a variant that plays a certain role in one context, and maybe the same exact variant has a different role in another context. And there are, lots of therapies that are approved variant-based or mutation-based in a certain gene in the context of one type of cancer and not in another type of cancer. I wonder if somebody wants to touch on what impact that, maybe Jenni, what impact that had on how different labs tiered those kinds of variants.
I think there's a really good example where we have most of our variant discordance in the solid tumor cases came actually in one particular VCF file. And that was in the context of a very mutated tumor because it was microsatellite unstable. And I think because of that context, when you look at multiple variants together, you come up with perhaps a slightly different interpretation than if you were looking at it as individual variants, as software often does. So that's one case where definitely the biological context of a tumour did impact the way it was interpreted. And I guess the other thing is we've seen examples where, laboratories, particularly with licensing of drugs in different contexts, would have slightly different tiering depending on the region they're located in as well. I mean, I know that you can build that into your software to a certain extent, but there are examples of, I know personally that, there's examples of drugs that are, approved in Europe, but not approved for use in countries because they won't be reimbursed by the individual bodies in those countries. And those kind of questions do impact on people's perception of tiering. I would say I don't think it should impact on tiering between tier 1 and tier 2, but I think the tier 1A and 1B, because if your reimbursement organisation doesn't consider it enough evidence to make it to reimburse for the costs, can you say that's a tier 1A? Is the evidence more that it would be a tier 1B? And that's, perhaps one way of looking at it.
Yeah, so that brings me also to kind of the specific case of hematological cancers. Sheryl, maybe you want to point out how differences in the diagnosis maybe make a difference in how people are tiering those variants.
Sure. I mean, the levels of evidence for prognosis and for prognostic and diagnostic significance is integrally related to the disease context. So how much evidence is there that this particular variant is associated with a negative prognosis in AML will be different from MDS or MPN or another kind of malignancy. And so the disease that it becomes critically important to be able to know what's the disease context of the variant, where sometimes you don't even know when you're collecting the sample what you're going to end up using the variants to help you with your diagnosis. So that becomes very complex when you have a variant that could be tier 1 prognostic in something like AML, but tier 2C in something else. Additionally, the specificity of the entered diagnosis, we found during the study that we found that if you entered an abbreviation, sometimes that could be misconstrued, and so the difference between myelofibrosis and mycosis fungoides led to a difference in tier assessment, and so that has since been addressed, but it's also been we didn't consider that to be an interpretation failing of the software either, because we're doing the interpretation within the context of the disease.
And I think it's worth saying that certainly our experts, a lot of them got very little clinical information when they're trying to classify the variants some of the time, or if they're trying to make the diagnosis. So that would also impact a little bit on that too. I mean, they just get it as a myeloid neoplasm without necessarily knowing all the clinical details of it. So that can be why sometimes they decided to apply a bit more of a general classification to things as a myeloid neoplasm rather than the specific disease. Right. And you can see why laboratories would do it in that way. It depends on how much information you have. I mean, somebody like Rich probably knows a huge amount about the patient details, but some of them don't have as close links to the clinicians that are, treating the patients.
So with these changes, I know that this study actually led to some updates and improvements in the bioinformatics tools, such as properly matching MF to the correct disease or giving a warning. There have also been changes in guidelines, et cetera. I wonder, what is the thought about this? This study was just published in June or July. It didn't finish very long ago. But if we were to redo it today, do you think we would get the same or different results?
I mean, I think we'd get different results, and I think there's multiple reasons for that. I think sometimes, as you've pointed out, the guidelines have changed. New clinical indications are coming on, new clinical trials being opened, which obviously changes the classification. From the point of view of the laboratories, I think the more experience they gain by doing their own classifications, I think they change the way they classify things as well, because it's still an emerging process, really, and an emerging field, and more information becomes available about variants. I think one example is there are new guidelines about hematological malignancies coming out, which put more stress on genes which previously were less important are becoming more important, or vice versa. And so I think we would definitely get different answers today. And in fact, we noticed that even within the study, because there had been a change. I think when we started the study, G12C variants in lung cancer were only in clinical trial. Agents against G12C variants in lung cancer were just in clinical trial rather than being approved therapy. And within the course of the study, they became approved therapy. So, the tiering of that changed within really the course of our study. So, that's quite an extreme example, because that was obviously quite a quick approval as well in that respect. But I wouldn't be surprised to see that happen more often.
Yeah, I would add that for variants, and that was an interesting one too, because KRAS variants are obviously well known and important, and the elevation of that one to tier 1A was an enormous deal. But then there are also variants that sit on that border between tier 2D or 2C, having a clinical trial that supports it, and not being a well-known variant, but not having any clinical relevance, which ends up with the current classification system putting it in a tier 3, like as a biologically relevant tier 3. And we saw that with APC over the course of the study, where it was originally a tier 2, and we reevaluated some evidence and it became a tier 3, and then a trial opened and it became a tier 2 again. So it's, that kind of, the variant itself hasn't changed, the impact on the disease hasn't changed, but the availability of a clinical trial or a piece of evidence has changed, and it changes the interpretation. And I think having a system that can contain that information, that's updated regularly, where you can rely on getting that information, can help maintain consistency across different laboratories that otherwise might not be aware of some of that information.
And one other thing, I don't think it's been mentioned yet, that I find interesting if you do these studies at different times is, our analysis and data about germline alleles as opposed to somatic alleles is evolving very rapidly also, right? And of course, that's going to affect, what gets included. If something was thought not to be a germline allele or variant, but actually, as we sequence more and more normal people, we see these come up, those databases are going to change as well, right? How often do we see a particular variant that might be a germline? Now, it's always not a problem in tumors, because the frequency of the mutation will often give it away. But we're seeing more and more mutations that we thought were purely somatic showing up in germline alleles. And what that means, I think, is still evolving.
Thanks.
I'd agree, Rich. And I think something that taps into that is we still have, variants in a particular gene where the genes of interest, we know the gene's relevant, but we don't know what that specific variant does. And that's, I think, also a really tricky area to resolve. Like, it's not just the variant, it's not just the gene, it's the variant, and the specific variants that we find that still end up as tier 3s because there is not enough evidence that they've got any functional effects yet. And that's, I find that's a very tricky thing to deal with still. I mean, I don't know. I think you've kind of alluded to that as well.
Oh, yeah. I mean, even in the laboratory, we struggle with this a lot, actually, right? Is that if you take one gene, and let's say the gene has 5 exons, and there are mutations in each exon, each of those could have biologically distinct and sometimes opposing functions, right? One exonic mutation could be oncogenic, the other could be tumor suppressive. It's a huge problem even in the lab.
We talk about those mutations all the time, and we like to call them hot threes, where it's something that it's not definitely benign, it's not definitely pathogenic, it's suspicious, but it's not really enough to call it likely pathogenic. It's more like a possibly pathogenic variant, and I think so much of that ends up being human judgment. If how much weight do you put on this particular biochemical assay, or has it been seen? If it's been seen in the germline, but maybe there's a biochemical assay that says it loses function, but there's a germline assay that doesn't associate it with disease. Does that mean it's just not fully penetrant, or it's not haploinsufficient? What do you do with that in a somatic context? It becomes very complicated, and so many factors like. How often has it been seen? Is it recurrent? What disease context is it in? And then pieces of information that we often don't have, like what is the phenotype of the patient?
Yeah, I gotta say, these are the ones that for me. Yeah, go ahead, Jenni.
Yeah, you might be thinking the same thing as me. And I think also as well is because if it's a therapeutic option as well, and it's something that we've not seen before, how do we know that it's going to respond to that particular therapy or not respond to that? I mean, I think it's a very difficult, and there's never going to be perfect answers, I think, for things where there's not enough evidence, is there?
Yeah, I was going to say, along the same lines, I think for me, these are the ones I struggle with the most, actually, right? Is that, the gene is important, and, let's say a particular variant's important in general, but then you have the mutation is 5 base pairs away in either direction. And these are the ones that kind of drive me nuts, right? Because you're like, it probably does something similar, but I don't know. Right? It might or might not. And those are the ones that I find I just wrangle with the most.
Right. And I guess this is where maybe this kind of second opinion can help weigh in and lean toward, how a physician or how the variant scientist is going to call those in the end. I think another point maybe to make is these also tier 3 variants that are in fact having an impact on gene function or on protein function, but there isn't, unfortunately, a therapy. Or maybe there's a trial, but not in your patient's region. How are you calling that? How are you putting that on the report, or are you?
And are you separating it in the report from all of the other tier 3 variants which have no known anything, which could be a very big list? And so then you as a user, as a reader of the report, as an oncologist, if you get this big list of tier 3s that includes both of those, how do you distinguish between them? Or should the report distinguish between them, even though they are, clearly tier 3 variants? I have an opinion, but.
So I wonder, kind of as a last question, because I think we have other questions from the audience that are coming in, which is wonderful. Jim, I guess I would like to direct this to you about what do you think are the benefits? Are there benefits? What are the benefits of clinical decision support software? Do they end up saving the user time? Are they to be trusted? Maybe kind of a leading question also in there would be, what's the role of automation or of artificial intelligence?
Sure. So I think, generally speaking, the question is like, how does this help, right? And it would be great if it was a super simple answer, but right, this is like clinical decision support software is a tool. It is the same, like if you think about it like that, this is a way to help a variant scientist, to help geneticists actually make better sense of what it is that they're looking at, right? You're getting bombarded with information. You need to stay on top of that information. And so the one question, does it save time? The answer sadly is yes and no, right? But there's good reasons to think about that. So from a yes, it saves time, you are able to go out and with this type of platform and others, scour so many different data sources and get a better understanding of what is out there and get a much more complete picture of the information available that you, as the person doing the interpretation, should consider. However, now that you're presented with all that information, you have a lot more thinking to do. And you have to figure out, how do I take all of these different data sources that didn't just show up in my normal Google search that I have perfected over the past couple of years, right? And so, yes, can it save time? Absolutely. Can it take a lot more time? Yeah, because you're getting a lot more stuff to look at, right? And so I think that's why, it's hard to kind of quantify how something like this would save time. But what it does come back to is an element of trust, right? I know that having used the platform and similar platforms, the one thing that would save time for me would be when I was actually trying to figure out what it was to look at. And so if I'm trusting the information in the knowledge base from human curated sources, from known databases, from previous reports, from expert summaries, if those are the kind of things that I know I can trust, that helps me at least speed up in the literature acquisition and the information retrieval side of it, right? And yeah, that's where I can trust those parts, and that will really help me at least feel better about the information that I'm using to really make a sense of all of this information that I'm using to interpret a variant. So it kind of goes across the board, right? And yes, there are different aspects for where AI could come into play, right? It could be something where how good is the platform at using these sort of computational methods and techniques to actually identify appropriate literature and to really kind of save time in the manual curation side of what do we actually have the curators look at, right? And are we there yet of how do we actually interpret a paper through the schema of a computer to tell you here's what's important? Not yet. Is it coming? Sure, it's really exciting. It's not there yet. But yeah, you can see avenues of where this could make my previous job as a variant scientist easier. And again, it is a tool. And this is something that we would use, and I have used, and how it can, help me make better sense of all of the information that's out there.
I think one of the things we found with the people that we talked to was that when they first encounter the tool, it's so transparent and everything is shown, like every piece of evidence. You can go dive into everything. And so you can go down as many rat holes as you want, but there is a proposed solution or answer for each thing. And there's, literature to support it. And there's, in many, 500,000 cases or more or less, there's a variant interpretation that's been written up by a scientist and supported with references that you can review. And if you want to go dive into each of those references, you have it right there to do it. But as you get used to how the tool works, you start to gain to know what you can trust and what you need to verify. And so ultimately, everything's presented to you in one place so you can evaluate it. But which things you need to evaluate, you sort of, as a user, learn what you trust and what you need to put your own judgment upon.
Yep, thanks. Yeah, and I think that Jenni mentioned earlier. I was just going to, go ahead, Jenni.
I think for us, from the way we look at, we're quality assessment assessing laboratories, and I think there is still that element. It's still a tool. You still have to use some judgment yourself as to whether you include things on your report or not. You have to speak to your local clinicians, and you just have to do a check sometimes to make sure that everything's sensible. Computers aren't perfect. People aren't perfect. So there's always it's a tool for us to use to aid in our variant interpretation. And I think, nobody's perfect.
I do think that what the tool can help you do is apply consistent standards and consistent rules that are being applied, so that while you do as an individual need to assess the validity of the evidence being applied, the rules that go into that classification are standardized, and so that can help alleviate some of the variability, although the individual pieces of evidence still are subject to different judgments.
Yeah, I would say
I think that's true, and I think it's...
Richard, why don't you go for that
one? One at a time. Jenni, you first. I was going to say, I'll just make a quick comment that I actually think that's true, that, it's kind of like the old line that your Google search is different than my Google search, right? And that I think if you don't have some level of uniformity in how you collate the data that's out there, some people are going to see some things and others are going to miss certain things. So I think actually having this first step of data curation through a program, I actually find to be tremendously helpful. I still have to go through the data that comes out the other side of it, right? But it eliminates the differences in how we might Google search vast amounts of VCF data and things like that. So.
Yeah, I would say, to follow up on that, like consistency, when I think about it, there are multiple aspects of where inconsistencies in human error can come in, right? And it really comes to people and across time, right? Those are really some of the two big things. So within an organization, within multiple organizations, if we're looking for consistency in interpretations, among multiple people looking at the same information at the same time, then yeah, like that's something where something like this can make sure that different groups have the same information, different people have the same information, and any sort of those algorithms that may be helping provide a classification to kind of help shape the point where you should be considering, that is consistent. But also the time thing is true too, right? As we were talking about before, new information comes out, and there needs to be the consistency in how the new information is interpreted in the context of what we now know. Right, and so those consistencies need to be, across groups, across people, and then time scale as well.
Yeah, and I think something also maybe important to point out is just like this particular tool combines this automated method with a person sitting behind it as well. Not that they're doing the full report. At the end, that's the end user's job, but it can provide confidence in what the output is and what the suggested call might be.
And I'm gonna add to that certainly we found with some of our discrepancies that they would be resolved by that, rather than the computer algorithm, by the person aspect of QCI1. So by submitting it for that extra level of interpretation by individuals. So I think that's a really valid way of doing things, because computers can't tell you everything, but you get better if you can send it off for a more curated individual human approach, and then the combination of both gave the best answer, really, I think it's fair to say.
Thanks.
Yeah, the computer has to make a decision, one or binary, 1 or 0 on on any given thing, and does and while individuals are assessing that data in the background for even the computed version, the computed classification is the computer, but having that additional second opinion that's incorporated within QCI1 allows the human to then review all the literature together and make an assessment that sometimes may differ from what the computer was able to come up with. So having both, I'm glad Jenni also agrees that was helpful.
Yeah. Okay. Thank you, everybody. Thank you, panelists. I think that we need to move now to questions from the audience.
Yes, thank you, Kathryn. I can take it from here. And thank you to all of our speakers for a very interesting and informative discussion. We will now start the Q&A portion of the webinar. To the audience, if you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located at the bottom of your screen. Let's get started. Okay, first question. How do you choose the variant allele frequency minimum and maximum values? Is it affected by the tumor percentage of the sample?
So, well, in this study, we set a cut-off of 5% because we had to set something up. But I think setting your cut-offs will depend on quite a number of different things within your laboratory setup, not least of which is whatever your limit of blank is. So you have to work out where there's noise in your system as well. So, I mean, the noise in the system is also variable depending on the samples, depending on, fixation artifacts and FFPE. You can get extra noise, which will affect the minimum variant allele frequency you want to set your cutoffs at. So, I mean, as much as you will set cutoffs, I think in your individual laboratory, there's still the best practice at the moment is to look at them, and there might be occasions where you want to move them up. And yes, I mean, the variant allele, the tumor content might have an impact, but it depends on what context you're looking at it as well. Because if you're looking at emerging variants in resistance or the like, they're not going to be necessarily at the highest variant allele frequency. They're not going to match the tumor content. And heterogeneity is something that people are very interested in. So it's not a simple trade-off, I don't think.
All right, we'll move on to the next question. Did you compare the interpretation from different labs using a uniform cutoff versus letting labs use their own threshold?
So we let the labs use their own thresholds, actually, for that one. Because we had to send them the VCF files, so the labs use their own cutoffs.
Our next question. Can divergent treatment decisions and outcomes around a variant allele frequency threshold be input into treatment algorithms post hoc to hone the guidance? And how would multiple algorithms be standardized so as to benefit from these data?
Rich, is this something that you can tackle?
Yeah, give me one second. I'm just reading the question more carefully.
Yeah. I guess one thing that comes to my
very
mind.
Interesting.
Okay. Go ahead.
You want me to continue, or you want to go? You can go, and then I'll pop in. Okay, I'll jump in. So
ahead,
let me just rephrase the question, because I think it's a very interesting question. Is if you set a VAF threshold, right? Can you later on go back and figure out? Perhaps maybe one way of saying it is, was there an ideal VAF threshold, right, that you look at post hoc? My short answer would be, I'm sure it can be done. It really is going to come numbers. Because what you need would be enough divergent outcome follow-up data to then say, okay, if we set the VAF here, this was the outcome. If we set it here, this was. I don't know that we have that type of bunch, but conceptually, it could be done, right? If we were to collect that, and Sheryl or somebody else could, but I don't think we're collecting outcomes after a VAF.
Yeah, I mean, I think there are certain cases like FLT3 in FLT3 ITD and in leukemia where you're really looking at the allele burden, and that makes a really big difference. That actually does make a difference in terms of treatment. I think in other cases it's not as clear. So I think it's going to really be ad hoc, like gene to gene, disease to disease, whether to what extent the VAF threshold is going to matter. It definitely does matter for FLT3 in leukemia, but I think that's really early data.
And I think the other thing is what makes that complicated is it's going to depend also where in their treatment course they are, because obviously if somebody has been treated with a set of agents, that could select for certain clones that have higher VAFs or lower VAFs. So you have to build that in as well, like not only what their disease is, but what have they been treated with and what type of clonal selection has gone on.
Right. And how many, if you have, where are they in their treatment? Like, have they had several lines of treatment? Is this a brand new variant? Even if it's at low frequency, is this the growing, you don't know which is the growing clone. So in a solid tumor where you're taking, especially if you're taking, circulating free DNA, it's very difficult to say in terms of.
Yeah,
like
that's
that,
what I was exactly what I was.
Go ahead.
Yeah, I was going to basically say the same exact thing, is that as we're collecting more circulating free DNA samples longitudinally now, when we're at MSK, we're sort of plugging all that data into our database. That'll be fascinating over time to see, can you reconstruct those evolutionary trees if you have enough data?
Right.
Thank you. We'll move on to the next question now. What was the depth for these variant calls, and has there been an established depth threshold?
Sure.
That's a good question. Jenni or Jim can probably speak to what we set in this study as a depth threshold. I know that that's something that different laboratories look at for sure when they're assessing whether or not a variant call is real. Whether there's a standardized threshold, I think that's kind of hard to say. Unless I'm
variant dependent as well.
Yeah.
Variant dependent, and I can't remember what we set. Jim, did we set a depth threshold?
I
can't remember if we did
for this. Yeah, I don't think there was a standardized one set for this. And again, this kind of goes back to some of the stuff we chatted about before, for just consistency and making sure like secondary analysis is easier than setting up your own scripting things now, but there still are a lot of opportunities to really tweak the output that you want and change the parameters according to how it functions in your own local setting.
Yeah,
I would have said that the primary bioinformatics would have done the depth, I think, more than what we did for this, but yeah.
All right, next question. Have you looked at the level of discrepancies in functional classification, i.e., whether the variant can cause cancer versus clinical classification?
In this study, Jenni, you can check me, but I think more of the discrepancy was at the clinical level, that there was largely very good agreement between QCI and the expert panel on what we would call pathogenicity or oncogenicity of the variant, the biological implications of the variant, and more discrepancy in the level of clinical implication. Is that fair?
I think that's fair, and I can only think of one case where it was more the biological that was the discrepancy rather than the clinical.
Yeah.
I think that there's only one variant, I think, that I can think of where that was the issue. So yeah, I would say that the study could look at both, but it was definitely, we saw more in the clinical and the biological.
Yeah, we did look at both, and I think there's something to be said about clinical decision support software in general, where QCI will err on the side of overcalling a variant in order to allow the user to look at it. Because if you're dealing with a really large panel or any kind of panel and you have a long list of tier 3 variants, it's going to be really hard to pick something that's important out of that big pile of tier 3s. Whereas if you have a short list of tier 1s and 2s and you think one really isn't significant and should be bumped down, it's much easier to look through that list and assess. So I think that it has to err on one side or the other, and that's where we've chosen to go. And I think that's the better way to go, personally, for that type of analysis.
All right, thank you. Next question. To what degree could variant classification be stable across scenarios versus context dependent? Generally, we accept that it should vary with diagnosis, but should it vary with other factors, for instance, presence of other variants, biallelic status, MSI status, etc.?
Yeah, so maybe I can just start briefly on this and pass it along. Just that I think, of course, variant interpretation is dependent on context. And fortunately, we can, QCI Interpret 1 can take co-occurring variant interpretation into account. So if you have biallelic, CVPA mutations, you can pull up guidelines that say, what impact that might have in the specific disease context. Or if you have a resistant mutation in the context of a activating fusion in lung cancer, versus a different cancer, that does need to get taken into account, I think. Maybe somebody else wants to follow up. Rich?
Yeah, I will. This one, context is dominant here. If you look at large-scale studies, right, that have assessed gene function, the dependency of a given cancer cell line is far more dependent on its lineage, meaning, what kind of cell it is, than the gene itself, actually. So from a broad perspective, context is actually probably more important than the gene itself in many cases. The problem is we don't have that much data, right? We don't have enough clinical data to always do that, so we have no choice but to generalize it, or as you said, sort of assume a certain stability across scenarios. But biologically, I don't think that's the case. And as we get more data, we're going to realize that context dependency probably kind of takes over in most cases, but we just don't have enough data yet.
All right, for the next question, this attendee specifically asked for Sheryl's opinion about variants with biological relevance versus variants with unknown significance.
I spend most of my brain on that topic. So in terms of reporting, I have a very strong opinion that they should be separated from the VUS list, because I think in the context of cancer, you never know when there's going to be a clinical trial. You never know. I think it's important that a physician see that those variants are present, even if there is nothing to specifically be done about them, because it could inform their understanding of how mutated a patient's cancer is, or how aggressive it's going to be, even if it's not doesn't reach a level of prognostic significance. I think it contributes certainly to the biology of the cancer, those variants, and I think those are very different from the novel variants that we don't know what they're doing, and so having them be separated within the report, I think, is important. How to interpret, and then so I think those are the biologically relevant tier 3s versus the not. Then there's the hot 3s, of course, which every single one is different. And within our team, we have daily discussions between variant scientists asking for second opinions. And sometimes we reach out to experts asking for second opinions. This is, at very, very low level in Nomad, but also high in Cosmic and right next to a hot spot, but no specific evidence. And those all fall into these possibly pathogenic, and everyone is different. I mean, we can set as many guidelines and write as many rules as we want in our curation manual, which we do, and we add to all the time. And we're still going to have edge cases that are difficult to assess. And then, each individual laboratory has a different set point where they feel that a hot 3 should be pushed over to pathogenic versus reported as a VUS, and different levels of conservatism in their own reporting. So it is a very challenging topic, probably the most challenging thing that we deal with.
Thank you. So it looks like we have time for one more question. Here it is. Are there any publicly available clinical decision support software, and what is the advantage of QIAGEN's versus those?
Is that one for me too? If my colleagues are aware of publicly available, I mean, there are certainly multiple different clinical decision support softwares available. I'm of course partial to the one on which I work, and I think there are a lot of advantages to it, including the large knowledge base that sits behind it, in addition to the people that sit behind it. And the combination of those, I think, are very powerful. There are other softwares out there that have other features. But I think I don't know if anyone else wants to weigh in.
I guess I would just say, you know, the combination, like we've talked about before, the combination of the classification that's pulling in curated information, making a computed classification, followed by expert interpretation makes, I think, QCI Interpret One a very powerful tool, better than. Well, I'm also very biased, right? I was a curator for this for a long time. It's high level information, and it's hard to beat.
And Katie and I know our team, so it's a team of very dedicated individuals.
And with that, thank you again, Jenni Fairley, Kathryn Bungartz, Sheryl Elkin, Richard White, and Jim Hayes for your time today and for your important work. Before we go, I'd like to thank the audience for joining us today and for your active participation. If you'd like more information, we invite you to check out the QIAGEN Digital Insights booth located in the exhibit hall. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speakers via the contact information you provided at the time of registration. I'd like to also thank our sponsor, QIAGEN, for sponsoring today's webinar. This webcast can be viewed on demand for 1 year until May 15th, 2025. Laboratories will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.